Hello students. So now we will be discussing about the acute immune system. So acute immune system, the immunity that we get after getting diseased or after getting encountered by some pathogens or some antigen. Okay, so antigens, these are the proteinaceous substance which are toxic in nature and which are released from the pathogens when they go inside our body. And for a particular antigen, our immune system, the very own immune system of our body, will produce a particular antibody. Okay, so let me show the structure of the antibody to you. Okay, so student, this is the structure of the antibody. Okay, this is the structure of the antibody. So antibody consists of one light chain. Okay, so there are two light chains. Okay, and there are two heavy chains. Okay, these are the heavy chains. Okay, so between the heavy chains there are disulfide bonds over there. Okay, and at the top of the antibody you will see some geometrical shape over there. Okay, so these geometrical shapes are specific to each antigen, and you can see from this diagram also the antigens are also showing some specific geometrical shapes. Okay, so these are the different geometrical shapes. So this, when this geometrical shapes, antigen and antibodies, they find each other and they will bond together, so there will be a antigen and antibody complex. So when this complex is formed inside our body, no disease will takes place, and if the antigen is present inside our body or has entered our body and it is not finding any specific antibody, so what will happen? The disease will cause. Okay. Okay, so the active and the passive immunity. So this is the difference table between the active and the passive immunity. So the passive immunity, the active immunity, sorry, it is developed due to the contact with the pathogen or its antigen. So whenever our body is getting encountered with the pathogen, so in response to that pathogen or the antigen, our body will be getting diseased, and as a result the infection is causing inside our body, the immune system, the immunity system of our body will produce the antibody. So it will take some time for the immune system to produce the antibody. Okay, so that is why it is very slow, but the process is very long lasting, student. Okay, so it is slow because it will take some time for the production of the antibody, but once the antibody is produced, so never and ever that specific antigen, if that antigen enter our body, it will not be successful in making the disease again. Okay, so now on the contrast, the passive immunity, it is developed when ready-made antibodies are injected into our body. So the ready-made antibodies we are getting from our mother when we are inside the mother's womb, and we are getting whenever we are transfused with the blood where the antibodies are there already. Okay, so as the antibodies are already over there, so that's mean it is very fast, it will prevent the disease very fastly, but it will only last for a few days. Okay, it is not a temporary process. Okay, because the antibodies which are present, okay, or which are injected ready-madely inside our body, so they are not the very own part of the human or any kind of animal's immune system. So the immune system of that organism will tend to reject that ready-made antibody. Okay, now one thing, the active immunity, here the antibodies are produced naturally after getting diseased. So the antibodies are the part of its own immune system, so there will be no side effect. But on the contrast, the passive immunity, over here they are the ready-made. Okay, so they may cause allergic reaction. Student, in this video we will learn how they can cause the allergic reaction. Okay, how being the not known part to our immune system, they can cause the allergic reaction. Okay, now it takes some time to response. Obviously the pathogen, the antibodies or the pathogen, in case of active immunity, they have gone inside our body. Okay, so it will take time for the production of the antibody, because the antibody will only be produced when the disease will takes place. Okay, but in case of passive immunity, it is a very instant process or response, because the ready-made antibody is getting injected into our body. Okay, so that is why it is spontaneous, and the antibiotic that we use, that we consume during the fever, okay, so these are also the passive immunity. Okay, so that immunity, that medicine you are taking, the antibiotics, that is for that time being only. Okay, so whenever your body is getting attacked or countered by some other bacteria, so again you will be suffering from the fever, so again you have to take some antibiotics, all these things. Okay, so the example of the active immunity is the vaccination, and passive is tetanus, all this thing. Okay. Okay, so now the principle of vaccination. So inside the vaccination, we generally use the small dose of pathogens or attenuated pathogens. Okay, so whenever you are injected with the vaccinations, so our body will get some symptoms of the disease. Okay, so for the time being we will be suffering from a mild fever or a chilled fever. Okay, so this is the reason of the primary immune response. Okay, so the disease takes place, okay, for some time. Okay, then after the production of the specific antibody, there will be no disease inside our body. Okay, but when the body is naturally getting infected with the pathogen, not in the form of vaccinations, so this is the natural infection, so the natural infection will be in higher dose, because in the vaccination we are intentionally we are using some smaller pathogens or attenuated means inactive pathogens. Okay, but in natural infection the nature will not do biasness towards us. Okay, so the nature will always consist of the strong pathogens, so when this pathogen will go inside us, our immune system will not show the primary response, that will show the secondary immune response. So they will be engaged themselves in the rapid production of the B and T cells. As a result, the antibodies will be formed later. Okay, and the response of the infection will be larger. Okay, so the disease will cause extremely. Okay, in case of natural infection. So student, this is the chart of various types of vaccinations. So normally in OPV we are using attenuated vaccinations, means attenuated pathogens, smaller dose of pathogens. In typhoid we are using killed pathogens. Tetanus we are using toxoids. Okay, we will be discussing what do you mean by toxoids. And in tetanus we are using prepared antibodies, that is why tetanus is a passive immunity, falls under the passive immunity. And hepatitis B, okay, so this is a second generation vaccination. So student, this thing we will be discussing in the biotechnology, whenever we will be discussing about the biotechnology chapter. Okay, now student, we will be discussing about the attenuated pathogens and the toxoid. Okay, so now see student, attenuated pathogen means where you have artificially reduced the potential of pathogenicity, that is the disease causing capacity or the potential of the pathogen to some lower extent, so that the pathogen will go inside your body but will not cause the infection or the disease to the extreme level, because you have reduced the potential of that pathogen. So you have reduced, student, this one, the virulence. Virulence is the disease causing capacity, potential. Okay, so you have reduced the potential of disease. inside the pathogen. So this is known as the attenuated pathogens. So sometimes the attenuated pathogens are also used inside the vaccinations. Okay, and the pathogens may also be inactivated by applying with the applying of the heat and the UV rays in case of some cancer disease, all these things. Okay, so we are also injected with the killed pathogens and the toxoid students. Okay, so now the immune system goes on the properties of the antigen and antibodies. Okay, so the antibodies special a particular antibodies are produced by knowing the structure, the geometrical configuration of the antigen. Okay, so if we have the information about the antigen, okay, what are the properties of the antigen, then we can easily can produce the antibody. Okay, so this is the main challenge for the scientists nowadays for who are finding the vaccination for the COVID-19. Okay, because the coronavirus, that is the COVID-19, is continuously mutating itself. Okay, so till now we have 3,300 different varieties of the COVID-19. So they are continuously mutating themselves, and as a result it's a very big challenge for the scientists to find out the right antibodies for that thing. Okay, so now in this case, in toxoid, you have the toxin. Okay, so you have removed all the components which are toxic in nature. So you are using a pathogen inside which you have removed all the pathogenicity from the pathogen. So that means the pathogen is now vacant. It only consists of the nature of the antigen, but the antigen will not cause any kind of disease. Okay, so this is known as the toxoid. Okay, so you have removed all the negative character from the toxin, so only the outer proteinaceous coat is remain over there, and our immune system has the capacity to translate all the characters of the antigen from that proteinaceous coat, and in response to that proteinaceous coat, our immune system will be able to produce a particular antibody. Okay, so this is the toxoid. Now see student, as I have told you that in case of passive immunity, sometimes the host, that is the in case of human beings and some other animals, they used to show some kind of allergic reaction, but in case of active immunity there is no side effect. Because why in passive immunity we see some kind of allergic reaction, I will explain you. Okay, so this is the case from where we are artificially ready-madely producing the antibodies. Okay, so for this we need the toxoid. Okay, so as we have studied now, the toxoid, this means only the proteinaceous coat of the pathogen, where the structure and the chemical properties of the antigens are present. They cannot produce any kind of disease or infection. Okay, so we have taken the toxoid tetanus. Okay, and we have put this toxoid into the horse body. Okay, so horse body injected, so the horse body is a living host, so they will produce the antibodies in response to this toxoid, because they have a structure of that antigen. So the immune system of the horse will read the structure of the toxoid, and in response to that they will be producing the antibody. Okay, so now time to time the blood from this horse will be collected, and inside the blood the ready-made antibody will be there, the antibodies of the horse. So these antibodies will be injected to the patient. In case of human beings, bigger animals, tiger, dog, cat, all these things. So now just use your common sense. Okay, so the antibodies of the horse, these are injected inside the other animals. Okay, so what will happen? So obviously the immune system, they know their own things. Okay, they can differentiate between the own things and the foreign things. So that is why whenever they are detecting the foreign things, okay, so obviously they will show some kind of allergic reactions. Okay, but the antibodies will do their job. Okay, so in this way they are causing the allergic reaction. Okay,